Bacterial growth. Bacteria are single-celled microbes. Bacteria reproduce by binary fission. In this process, the bacterium, which is a single cell, divides into two identical daughter cells. Binary fission begins when the DNA of the bacterium divides into two replicates. The bacterial cell then elongates and splits into two daughter cells, each with identical DNA to the parent cell. Each daughter cell is a clone of the parent cell. When conditions are favorable, such as the right temperature and nutrients are available, some bacteria-like can divide every 20 minutes. This means that in just 7 hours one bacterium can generate 2,097,152 bacteria. After one more hour, the number of bacteria will have risen to a colossal 16,777,216. That's why we can quickly become ill when pathogenic microbes invade our bodies. When grown in culture, a predictable pattern of growth in a bacterial population occurs. This pattern can be graphically represented as the number of living cells in a population over time and is known as a bacterial growth curve. Bacterial growth cycles in a growth curve consist of 4 phases: lag, exponential, stationary, and death. The bacterial growth curve represents the number of live cells in a bacterial population over a period of time. Lag phase. This initial phase is characterized by cellular activity but not growth. A small group of cells are placed in a nutrient-rich medium that allows them to synthesize proteins and other molecules necessary for replication. These cells increase in size, but no cell division occurs in the phase. Exponential phase. After the lag phase, bacterial cells enter the exponential phase. This is the time when the cells are dividing by binary fission and doubling in numbers after each generation time. Metabolic activity is high as DNA, RNA, cell wall components, and other substances necessary for growth are generated for division. It is in this growth phase that antibiotics and disinfectants are most effective, as these substances typically target bacteria cell walls or the protein synthesis processes of DNA transcription and RNA translation. Stationary phase. Eventually, the population growth experienced in the exponential phase begins to decline as the available nutrients become depleted and waste products start to accumulate. Bacterial cell growth reaches a plateau, or stationary phase, where the number of dividing cells equal the number of dying cells. This results in no overall population growth. Under the less favorable conditions, competition for nutrients increases and the cells become less metabolically active. Spore-forming bacteria produce endospores in this phase, and pathogenic bacteria begin to generate virulence factors that help them survive harsh conditions and consequently cause disease. Death phase. As nutrients become less available and waste products increase, the number of dying cells continues to rise. In the death phase, the number of living cells decreases exponentially and population growth experiences a sharp decline. As dying cells lyse or break open, they spill their contents into the environment, making these nutrients available to other bacteria. This helps spore-producing bacteria to survive long enough for spore production. Spores are able to survive the harsh conditions of the death phase and become growing bacteria when placed in an environment that supports life. Bacteria, like all living organisms, require an environment that is suitable for growth. This environment must meet several different factors that support bacterial growth. Such factors include oxygen, pH, temperature, and light requirements. Each of these factors may be different for different bacteria and limit the types of microbes that populate a particular environment. Bacteria can be categorized based on their oxygen requirement or tolerance levels. Bacteria that cannot survive without oxygen are known as obligate aerobes. These microbes are dependent upon oxygen, as they convert oxygen to energy during cellular respiration. Unlike bacteria that require oxygen, other bacteria cannot live in its presence. These microbes are called obligate anaerobes, and their metabolic processes for energy production are halted in the presence of oxygen. Other bacteria are facultative anaerobes and can grow with or without oxygen. In the absence of oxygen, they utilize either fermentation or anaerobic respiration for energy production. Aerotolerant anaerobes utilize anaerobic respiration but are not harmed in the presence of oxygen. Microaerophilic bacteria require oxygen but only grow where oxygen concentration levels are low. Campylobacter jejuni is an example of a microaerophilic bacterium that lives in the digestive tract of animals and is a major cause of foodborne illness in humans. Another important factor for bacterial growth is pH. Acidic environments have pH values that are less than 7, neutral environments have values at or near 7, and basic environments have pH values greater than 7. Bacteria that are acidophiles thrive in areas where the pH is less than 5, with an optimal growth value close to a pH of 3. These microbes can be found in locations such as hot springs and in the human body in acidic areas such as the vagina. The majority of bacteria grow best in sites with pH values close to 7. Temperature is another important factor for bacterial growth. Different bacteria grow at different adaptive temperatures. Some bacteria can adapt to temperatures below 4 degrees Celsius or even below 0 degrees Celsius. Generally, bacteria living in humans can adapt to temperatures of 20 to 45 degrees Celsius. Growth was best at 37 degrees Celsius or near body temperature. There are also bacteria that can withstand temperatures of 50 to 80 degrees Celsius, which can be found in hot springs and geothermal soils. Some bacteria require light for growth. These microbes have light-capturing pigments that are able to gather light energy at certain wavelengths and convert it to chemical energy. Cyanobacteria are examples of photoautotrophs that require light for photosynthesis. These microbes contain the pigment chlorophyll for light absorption and oxygen production through photosynthesis. Other bacteria, such as purple and green bacteria, do not produce oxygen and utilize sulfide or sulfur for photosynthesis. These bacteria contain bacteria chlorophyll, a pigment capable of absorbing shorter wavelengths of light than chlorophyll. Purple and green bacteria inhabit deep aquatic zones.